Okay, in the previous lecture, I talked about transcriptional regulation primarily in the eukaryotes. I, of course, did not get into much of a detail. We did not talk about all sorts of operons. I certainly omitted fudge genetics, which is a very, very critical aspect. I would encourage all of you to read more about it. Uh, there are some fundamental insight that we got from fudge genetics, which is used, which was used again when we uncovered regulations of eukaryotic transcription. But nonetheless, um, we will try to get in quite some depth when it comes to eukaryotic transcription. See, the basic mechanism still remains the same. That is polymerization of RNTPs into RNA molecules using DNA as a template. The process involves determination of the start site for transcription and the stop site for transcription. That is, which part of the DNA will be copied into an RNA. Okay. However, the details of it, the regulation of it, is mind-bogglingly more complex. First of all, you think about how the chromatin is condensed. It is condensed in several tiers that you all know about. Then, the DNA, the chromatin itself can be condensed or not, depending on the status of acetylation and methylation of the histones. Acetylations open it up, methylations close it down, make it more condensed, and so on and so forth. Then there are, proteins that are recruited to the chromatin specifically because of these chemical modifications, acetylation, methylation, those are there. But also the basic mechanism is a whole lot more complex. Here there are several general transcription factors, what is shown in this on this slide are general transcription factors for RNA polymerase II. So they are written as TF2A, TF2B, C, D, E, F, H, what's are. And then there is actual RNA polymerase, which has a whole lot more subunits than the prokaryotic counterpart. Then there is a ginormous complex called the mediator complex, which plays a very critical role in transcriptional regulation. And then there are these tissue-specific transcription factors, or situation-specific transcription factors. So all of these are involved. Then there are regulations at the elongation stage as well. I do not focus on the elongation stage much, even the worldwide research, if you think about, it is more focused on the initiation stage than on the elongation stage, for a simple thing that because polymerization of RNA or transcription is such energy intensive process that evolutionarily majority of the regulations are in the initiation stage. Less is there in the elongation stage. There are instances, but primarily it is in the initiation stage. So we will focus primarily on transcription initiation. Like when we talk about transcriptional control, we talk about transcription initiation. So this is the this one slide. I will take today's whole lecture just to explain this one slide. First, let us start with the similarities between prokaryotic and eukaryotic transcription initiation. Okay, both have a Tata box-like feature. Okay, so this is the promoter element, which is rich in TAs and As, and that's where the RNA polymerase is recruited. Both have a sigma subunit-like feature. In prokaryotic transcription, the RNA polymerase comes with it. The sigma subunit is considered to be part of RNA polymerase, though it's a metastable association, meaning sigma subunit remains associated with the RNA polymerase only during the the initiation part, and during elongation, it is not part of it. Okay, but in eukaryotes, there is a completely different general transcription factor called TF2D. Both sigma subunit and TF2D, they are the proteins that actually make contact with the promoter, the Tata box. Okay, so yeah, like there is not exact similarity, like sigma subunit is part of RNA polymerase II in prokaryotes, while in eukaryotes, TF2D is it's a separate protein complex altogether. It is not part of the RNA polymerase, but it performs the same function. Then they have quite similar structure, the overall architecture, the saddle-like architecture, they both have. Okay, then yeah, the RNA polymerase subunits 1 and 2 are similar to bacterial beta prime subunits. Let me tell you a little bit about how these subunits are named. You see, it is RPB1 and RPB2. RP is RNA polymerase. B is alternatively used as 2. As you know, that mRNA transcription is done by RNA polymerase 2. In eukaryotes, there are at least 3 polymerases. Now people say there are 5 polymerases at least. But RNA polymerase 2 is the one that transcribes mRNAs, which is the focus of this series of lectures. So alternatively, it is called RPB. Okay, so RPB1 and RPB2 means subunit 1 and subunit 2 of RNA polymerase 2. They are very similar to bacterial beta and beta prime subunits. RPB3 and RPB11 are very similar to bacterial alpha subunits. Now, what are the differences between prokaryotic and eukaryotic transcription? Differences are way too many. Okay, in eukaryotes, there are 3 RNA polymerases. RNA polymerase 1, it is located in the nucleolus and transcribes genes for ribosomal RNA. RNA polymerase 2 is located outside of the nucleus and it transcribes the protein coding genes. RNA polymerase 3 is located outside the nucleolus in the nucleus and it transcribes one ribosomal RNA gene and all the tRNAs. Okay, and now in plant they say that there is RNA polymerase 4 and RNA polymerase 5 as well. Okay, but like we will not talk about them. In fact, we will not even talk about much about RNA poly 1 or RNA poly 3. This is very important, that unlike in the prokaryotes, in eukaryotes, RNA polymerase cannot bind to the promoter on its own. Unlike in the prokaryotes, in the eukaryotes, RNA polymerase cannot bind to the promoter on its own. Please remember that in prokaryotes, sigma subunit is part of the RNA polymerase. Okay, how was this demonstrated? This was demonstrated by a classic series of experiments conducted in the laboratory of Robert Freuder in Rockefeller University. What did he do? He, like many groups before him have shown that, you know, like you can do transcription in vitro, where you simply take a known piece of DNA which will have a promoter, you add nuclear extract to it, then and radioactive RNTPs, you will get production of radioactive RNA. Okay, polymerization of radioactive RNTPs can happen in vitro. So nuclear extract, why? Because transcription is a nuclear event, right? So if you take nuclear extract from human cells, it can support transcription from known promoters. Okay. However, if you purify the RNA polymerase II, then that cannot transcribe from the known promoters. How was it demonstrated? That if you take a piece of DNA and if you know exactly where the promoter is, And because it's a linear piece of DNA, so you know that this is where it ends. Okay, it's a restriction fragment, right? So if you know the promoter, around 25 to 30 nucleotide is where the transcription will start. If you know exactly up to where it is, then you can you know that the transcript size will be 800 base pairs. Okay. If you take pure RNA polymerase II and a piece of double-stranded DNA that contains a promoter, then you will see that the product is of very light intensity and all kinds of sizes. So the conclusion is that pure RNA polymerase II cannot initiate transcription from the promoter, but it can initiate non-specific transcription from a template. Non-specific matlab, like it may be from the promoter, it may be from before the promoter, from after the promoter. There is no specificity in the transcription start site. Remember, this is the thing about transcription, that the process of transcription and transcription regulation is to determine from where the transcription will start and from where it will end. Okay, so if you take a pure RNA pol II, it can initiate transcription weakly. It is not a very strong transcription, but non-specific. Okay, so therefore Bob Roeder deduced that if pure RNA pol II cannot initiate transcription from the promoter, then it must require some other factors, and those factors must be present in the crude extract here, and that became the basis of the assay for the purification of transcription factors that are associated with pol II, RNA pol II. How would you do that? Simple. You take the pure DNA template where you know the size of the transcript if it is starting from the promoter. You add pure RNA pol II to it, add radioactive RNTPs and buffer, and you will not get specific transcription. Right? You will run on a gel and you will get this kind of a thing. Then you start to fractionate it. Okay, so you take a guess that because this will bind to RNA, therefore they bind to DNA, therefore the protein might be positively charged. So you use a negatively charged column called phosphocellulose, and it is possible that the transcription factors which are present in the nuclear extract might bind to the column. Then you elute it, and then you do it again, and then you might start to see a band. That is a radioactive band of RNA, which can result only from specific transcription initiation from the promoter. Because now the RNA pol II cannot bind before or after the promoter. It has to bind here only, and therefore you will get a defined size product. But in one step the protein will be not pure, so you will do size exclusion chromatography, you do other kinds of chromatography till you get a pure protein solution. And that's how it was done. And in the final step, there was, this is the original paper from Bob Roeder's group. And in the final step, the proteins were bound to phosphocellulose column or an cation exchange column, and depending on how tightly it is bound to the column, some proteins came out first, at a buffer with a buffer that has relatively low salt concentration, and some proteins came out much later. I do understand that you may not have first-hand experience of protein purification. Generally speaking, we use 100 millimolar potassium chloride in the buffer. That is the salt concentration we use. It is almost the same as our body, okay, isotonic solution. When you are trying to elute proteins, you increase the salt concentration, maybe 150 millimolar, maybe 200 millimolar. So TF2A came out first, meaning it has minimum affinity towards a negatively charged column, phosphocellulose column. You keep on increasing the, you get TF2B, TF2C. TF2D has so high concentration that you require 1 molar salt concentration. 1 molar. That's a whole lot of salt concentration. You will rarely find examples where such high salt concentration is used to elute a protein out of a chromatographic column. The reason is that, remember, TF2D is the transcription factor that binds to the DNA. Okay, the promoter, that binds to the promoter. So this is the transcription factor that makes direct contact with the promoter. Therefore, it has the highest affinity towards, DNA or DNA-like substrate, which is the phosphocellulose column. So it came out last. So that's how they were named, TF2A, B, C, D. But later on, other general transcription factors were also found. Please refer to the last lecture. These are the transcription factors that are called general transcription factors, or GTF. Okay, these are required for transcription of any mRNA. That's why they are general transcription factors. Now, if you look at it, then just like in the prokaryotes, you see that here also there is a kink in the DNA. This is the promoter. That's where the RNA pol 2 is recruited. And see, TF2D, it has this kind of a saddle-like shape of a particular subunit of TF2D called TBP, Tata binding protein. Okay, this is TBP, Tata binding protein. There are other proteins. So TF2D is equal to TBP plus Tata associated factors. Okay, it's a multi-subunit protein complex. So there are multiple Tata associated factors. This complex interacts with what is called mediator. We will explain mediator a little later. A mediator complex, and which in turn interacts with these proteins, which are your tissue-specific transcription factors. They bind to the part of the DNA which is called the enhancer. Okay, we will come to all of this. We will define everything. So, and you see that these tissue-specific transcription factors, almost always, they have this feature that they have a DNA binding domain, which will dictate the sequence specificity with which they will bind to the DNA. Just like restriction enzymes, they can only recognize a particular sequence and cut, right? So these DNA binding domains will dictate exactly which sequence on the DNA that the transcription factor will bind to. And they have an activation domain. Sometimes this can be repression domain as well. This activation domain of transcription factors, every time I say transcription factor, I mean tissue-specific transcription factors. If I want to mean these TF2A, B, C, D, etc., I will specifically mention GTFs. Okay, so these transcription factors, they will have a DNA binding domain, they will have a transactivation domain, and this DNA binding domain will have different kinds of specificities that will dictate which sequence they will bind to. But the transactivation domain, almost without a miss, they will interact with the mediator complex. And these are called domains. By definition, domains are structurally and functionally independent. So that's how they are shown here. One blue ellipse, one green ellipse, and a little flexible region in between. So technically speaking, you can take DNA activation domain of one protein, one transcription factor, and swap it with the activation domain of another transcription factor. The new hybrid transcription factor will also act exactly like the transcription factor whose DNA binding domain you are using. Or you can take, so that's how it is. So you see that this is the RNA pol 2 and this is the mediator complex that interacts with it. This is the yeast mediator complex. The human mediator complex is also very, very complex. There are way too many subunits. Some of these mediator complex have properties of chemical modification of the chromatin. Some of them have a property of interacting with the RNA pol 2, some with general transcription factors, some with activated like tissue-specific transcription factors, and so on and so forth. So the eukaryotic transcription machinery, therefore, is mind-bogglingly complex. Okay, it has like many components, and then on top of that, there is a special feature. The largest subunit of RNA pol 2, RPB1, it has a C-terminal domain which has a unique sequence. It's called a heptad repeat. Heptad meaning 7 amino acids. So 7, 1, 2, 3, 4, 5, 6, 7. And look at the amino acids. Tyrosine, serine, forget proline, threonine, serine, forget proline, serine. All of these can be, sorry, not this one. All of these can be phosphorylated. Okay? Phosphorylation means you will be imparting negative charge to the polymerase, and that will weaken potentially its interaction with the DNA, because DNA is also negatively charged. That's number one. Number two, as you are changing the charge property of RNA polymerase 2, it may acquire property of interacting with some other protein. See, like when you are changing the charge property, it is not just you are changing the charge property. It can also change the structure of the RNA polymerase. So both will happen. The charge and the structure of RNA polymerase can change based on this post-translational modification. Okay, the charge property, like the phosphorylation. So you will see the importance of that, in the next few slides, and again in another lecture when I will talk about post-transcriptional regulation of regulation in eukaryotes. Okay, so depending on the status of phosphorylation, it can acquire the property to escape the promoter clearance. You saw that happens in bacteria as well. Soon after the promoter clearance, the RNA, the 5' end, will try to peep out of the RNA polymerase. When that happens, then it needs to be capped. Okay, the capping, the 5' capping of the nascent mRNA happens like that. So depending on the phosphorylation status, then it will acquire the property of interacting with the protein complex that can do the capping. Okay, the capping enzymes. Then when the phosphorylation property will change further, it has moved down the DNA a little bit more RNA has come out. It will then acquire the property of recruiting some of the splicing machinery, so that splicing can happen. So phosphorylation of this C-terminal domain, therefore, is a very critical thing. And see, like this is said that it has an essential carboxy-terminal repeat. This essential part I will explain to you in a little later slide. And there is a species-specific variation. The C-terminal domain of yeast has 26 repeats, while in mammalian it has 52 repeats, and all sorts of stuff. Okay, so yeah. So the phosphorylation is primarily in the serine threonine, and some tyrosine residues can also get phosphorylated. Okay. You can actually see it on a DNA. If you take the polytene chromosome of Drosophila, which is, multiple copies of the same sequence of DNA. So if you are trying to see some DNA-based events, this is a very good system. In fact, DNA banding, etc., are all discovered in polytene chromosomes. If you do that, and if you take two different antibodies, one antibody that is specific for phosphorylated CTD, it is fluorescently tagged with red color, and another antibody that is specific for the unphosphorylated CTD, and it is fluorescently tagged with the green color. You can see that certain parts of the chromatin is green, certain parts are red, and certain parts are hazy yellow, and stuff like that. And these are, very actively elongating RNA polymerases there. Okay, so this is what it is. Okay, see, this RNA polymerase phosphorylation status is very critical for eukaryotic transcription. So what are the things that are there? In prokaryotes, you have a core polymerase, sigma factor. In some cases, you have proteins that bind to the operator. We have also see the inertic protein that acts like a gene-specific transcription factor, something like that. But in eukaryotes, the core polymerase has 12 subunits. Then there are GTFs, which does not even exist in prokaryotes, at least not that we know of. There is a multi-subunit mediator complex. Then there is Swiss-Nif complex. Then there is a wide array of activators, repressors, etc. And then there is a DNA element called enhancer. Enhancer is very similar to what we learned in the last class. Like if we go back there, like this NTRC, that it bound to this sequence and it interacted with the RNA pol bound to the promoter. That is called an enhancer sequence. Okay, and that is standard for eukaryotic genes. Okay, so this is a cis element. Everything else other than the enhancer, everything else is called trans elements. Enhancer is called cis element. So the cis and trans is a very commonly used term in biology. It depends on what is it that you are calling the system. Okay, so if in this case, because transcription happens using DNA as the template, so DNA is the system. So anything that is integral part of the system is the cis element. Anything else that is coming from outside is the trans element. So all the protein factors, GTFs, RNA polymers, etc., all are trans factors. Okay. Then the nascent RNA is post-transcriptionally modified. It is capped, it is polyadenylated, it is spliced, and for all of these, RNA polymerase post-translational modification status is important. And finally, transcription and translation are uncoupled. In prokaryotes, transcription and translation happens simultaneously, but in eukaryotes, transcription happens in the nucleus, then it comes out in the cytoplasm. That's where translation happens, right? So this is taken from a review article. I would really encourage you to read this review article. It is very, very good. Okay, this just talks about the status of RNA polymerase to phosphorylation and how that coordinates different activities along, transcription, initiation, and then elongation, and then the post-transcriptional modification events. Okay, so this is very, very interesting. You can read the review article, easy to read, and you can read it. Now, I am coming to a very important philosophical understanding for all of you. Okay, so you see that often in textbooks, in colloquialism, you say or you have heard or you have read that the reporter gene is fused downstream of the promoter of that gene. Okay. Now I will try to establish to you that why that is such a wrong thing to say. Okay. So we know that in humans, roughly 23,000 different mRNAs are encoded, right? In human genome, 23,000 different mRNAs are encoded. And in a particular, see the fact that your skin cells are very different from your muscle cells, are very different from your bone cells, which are very different from your neurons, tells you that if all the genes were transcribed and translated in all the cells, then all the cells would have looked alike, the same. But that's not the case. Neither do they have the same morphology, neither do they have the same physiology. So therefore, all the genes are not transcribed and translated in all the cells. Rough estimate is that each distinct cell type transcribes and translates roughly 5,000 different genes. So if therefore 23,000 is your superset, 5,000 is a subset, and that subset is kidney specific. Like kidney itself has 80 different cell types. Okay, so one of the kidney cell type specific subset of 5,000, another kidney specific cell type subset of 5,000, bone-specific cell type of 5,000, the bone-specific subset of 5,000, neuron-specific subset of 5,000, and there are variations even within that. Like the total number of cell types, we have no clue. Like I would think that we have thousands of different cell types, which are all molecularly distinct. Okay, nowadays that there are a lot of this single cell transcriptomic analysis is being done, which is clearly elucidating that the number of different cell types that we have thought to exist before is clearly a gross underestimation. If you go by molecular criteria, as in the genes that are being transcribed in a particular cell type, the number of different cell types is much higher than what we estimated earlier. But almost, I'm saying almost because in the next slide I have to tell you about the exception, but almost every gene that is transcribed will have a promoter, right? Every protein coding gene that is transcribed will have a promoter. I am being very specific. Protein coding gene that are transcribed will have a promoter. And if I ask you, or any one of you, that tell me the promoter sequence, you will be able to tell me. You will say that it is, TA, TAA, something like that. In fact, if you align the promoter sequence of thousands of different genes, you will see that there is remarkable conservation in the sequence. The first base, being a T, is correct in 83% cases. The second base, being an A, is correct in 91% cases. The third base, being a T, is correct for 100% cases. The fourth base, being an A, is correct for 95% cases. So you see that the promoter sequence, the Tata box, which is roughly 25 to 35 nucleotide away from the transcription start site, that sequence is more or less invariant. It's the same sequence for every gene. So if the sequence is invariant, how can it bring about variation in transcription? Meaning, how can the promoter sequence dictate that this gene will be transcribed in liver but not in brain? That gene will be transcribed only in adult muscle but not in muscle progenitors. This variation cannot come from something that is invariant. And that is the reason why I say that do not say that I will attach the reporter to the promoter of the gene and I will see where it is expressed. Promoter cannot dictate the variation. The variation is dictated by enhancer. Okay, so again, I will start with the concept that I started talking about earlier on, and I will keep on talking about it. If you consider RNA polymerization of RNTPs as a chemical process, for which the necessary precondition is that the RNA polymerase have to be recruited to the promoter. Only then it can happen, right? That we have decided, like already understood. Now, it is the promoter sequence is the same. So, if it is not being transcribed in liver, meaning that despite having the promoter sequence and the presence of the RNA polymerase and all the GTFs, RNA polymerase is not getting recruited to the promoter. So something else is dictating the rate of recruitment of RNA polymerase to the promoter. And that something else is your transcription factors and the enhancer sequence to which it binds to. That enhancer sequence is very different from gene to gene. So the variability comes from the enhancer. So you should always therefore talk about promoter enhancer, not just promoter. Yeah? I hope I could make myself clear. But while I almost made promoter the unitary god of transcription, it is not exactly true. There are some genes, it was discovered by Richard Young and Kevin Struhl almost simultaneously around year 98, 99, that there are certain promoters that are like certain genes for which the conventional Tata box promoter is not there. Rather, there is something called an initiator. The initiator sequence is very different. Okay, so the initiator sequence is not that well defined as the promoter sequence. Like as they say, there is a C at -1 position and an A at +1 position, and like the sequences around it defines how one particular sequence will act as a initiator sequence, which is very different from the promoter. In promoter, the sequence between the Tata box and the transcription start site, this sequence really doesn't matter. You can change it in every which way and transcription will happen just like that. But in initiator, the surrounding sequence is very, very important. The initiator sequence per se is very degenerate, but the surrounding sequence is very, very important. Okay. Now comes the mediator complex. There is an interesting history of discovery of the mediator complex. See, when I talked about Bob Roeder's experiment that you take a double-stranded DNA, you add RNA pol2, and you see whether transcription is happening or not, that was the norm between late 70s to early 90s. That was the norm. That everybody would do these assays, in vitro transcription assays, on naked DNA. What is naked DNA? You all know, right? The DNA in the cell, whether it is prokaryotes or eukaryotes, you know particularly well about eukaryotes, that it is wrapped around a nucleosome. So DNA inside the cell is never pure DNA, okay, naked DNA. DNA inside the cell is in the context of nucleosome. So the moment, so these in vitro assays therefore were a slightly artifactual system. The moment the naked DNA was replaced by DNA that is wrapped around nucleosome, then pol2 and GTFs could not initiate transcription. Okay, see, like when Bob Roeder did his experiments, he showed that you take naked DNA, just pol2 cannot transcribe specifically, very weak transcription, non-specific transcription. You add GTFs, you get a lot of transcription, specific length. But that was naked DNA. That was an artifactual system. The moment you wrap the DNA in nucleosome, Bob Roeder's experiment is not replicatable. Then you see that pol2 and GTF cannot initiate transcription. So now you have a new assay, right? So far, you purified the GTFs using the naked DNA based assay. Now you see that in the nucleosomal context, that transcription cannot happen with pol2 and GTF, but nonetheless, transcription happens inside the cell, and it happens when there is nucleosome. So what Roger Kornberg started to do is he took the nucleosomal DNA and gave pol2 and GTFs and see there is no transcription, but then he added nuclear extract to it and he could see transcription. Then he started purification and purified a complex that is 20 subunit protein complex that could support transcription of DNA in the nucleosomal context in vitro. Okay, so that he named as the mediator complex. And you know what, quite surprisingly, when he did that, when he sequenced those subunits of the mediator complex, he realized that most of these proteins were already known. They were known in certain yeast genetic screens. So these proteins therefore have very unique names. Okay, look at it. So as I told you that the RNA pol2 is also referred to as SRB. Okay, RPB. Okay, so I will show you how. So just remember these names: SRBs, SWI/SNPs, and SRBs and SWI/SNPs. Okay, and these are part of the mediator complex, and I'll tell you how those genes were discovered. SRBs. You remember that we talked about the heptad repeat, and I highlighted the fact that this is essential repeat. If you make an yeast strain where the RNA pol 2 RPB1 has everything, but the heptad repeat is shortened, okay, then the yeast cell was sick, it could not grow well, okay. So then you take that kind of a cell and like either mutate them or do library-based overexpression assay to look for suppressors. That is there any genetic change that can suppress this slow growth phenotype resulting out of deletion of the C-terminal heptad repeat. So therefore, these are suppressors of RNA pol B. Actually, RNA pol B heptad repeat mutation. So these SRBs were discovered and they were known that these proteins have something to do with the heptad repeat part of RNA pol 2. Roger Kornberg identified those, he used a biochemical approach. When he sequenced it, he realized that some of the subunits of mediator complex are basically SRBs. Okay. Ida Hertzkowitz, she was doing something completely different. Okay, that yeast has two mating types, right? The A type and alpha type. If we get an opportunity, I will talk about that. It's a very, very unique thing. Like I do not know, like I'm really old and you guys are much, much younger. If you have ever seen a radio cassette, like a audio cassette player, I don't know if you have ever seen it. So you can have a whole stack of cassettes in your rack, but only the cassette that is within the cassette player and you turn it on, only that music will play, right? So there are two copies of, like there are two loci, okay? One here, one here. One is the alpha loci, one is the A loci. Both of these are transcriptionally inactive. But there is a nuclease called, I do not remember the full form of HO, this HO nuclease that can cut this part and insert it here. This is the active part. This is your audio cassette player. So whichever is inserted here will be transcribed and the mating type will change. So it's mating type switch. Okay. So the switch gene means switch. Okay. So they were assaying for genes which, if mutated, this mating type switch will not happen. Now, this mating type switch involves active transcription. There is some transcriptional regulation is involved there. Okay. So some of those mutants that Ida Hertzkowitz identified and the corresponding proteins, those were also subunits of the mediator complex. Similarly, I gave you this paper or a variant of this paper as part of your quiz, that you take yeast cells and assay for genes which, if mutated, yeast will not be able to grow in presence of sucrose as the only carbon source. So those mutants are called sucrose non-fermenting, SNF. Now, for yeast cells to grow in presence of sucrose as the only carbon source, minimally they will have to turn on the transcription of invertase, the enzyme that is secreted and degrades sucrose into glucose plus lactose, and then glucose is taken in, right? So in all the three cases, like SWI and SNF, switch and SNF, okay, these are specific cellular events where selective transcription or transcriptional induction is critically important. Okay, and there were genes which, if mutated, this process is blocked. And SRB mutants were those which were somehow interacting with the phosphorylated heptad repeat. So you can imagine that all of these therefore have something to do with activated transcription. And that's what was lacking. When there was in vitro transcription with naked DNA, you were just measuring the basal rate of transcription from a naked DNA. The moment you put it in nucleosomal context, it was not sufficient. So you then need transcriptional activation. And for that, therefore, mediator is an essential component. So the bottom line of saying all of these is that for activated transcription, you need mediator complex. So when Roger Kornberg's group, they purified the mediator complex, they realized that when they purified it, they found that SRB 8, 9, 10, and 11 were never present in the mediator complex that they isolated. Now, see the interesting aspect that SRBs are somehow involved with interaction with RNA-pol2. And many of the SRBs are involved in activated transcription, but these 4 SRBs were excluded. Now think of the assay that Roger Kornberg was using. What is it? That there is no transcription. You add the mediator complex, you get transcription, right? So this is a transcriptional activation assay. Is it possible that these SRBs are not involved in activation of transcription, but in repression of transcription? Now think back in bacteria, tryptophan operon is a repressible operon where the default state is on, but when you have a lot of tryptophan, then it is repressed. While lac operon is an inducible operon where the default state is off, only when you need it, it is turned on. So that will be the case with the SNF mutants. They must be involved in activation of transcription, right? So these 4 mediator SRBs were missing in the mediator complex that Roger Kornberg first isolated. So you see, like, this is how the arrangement is, that this is the promoter where the RNA-pol2 is there, all the GTFs are there, and the mediator complex is the reason it is called a mediator complex. It mediates the interaction between tissue-specific transcription factor and RNA-pol2. And this tissue-specific transcription factor is bound to the enhancer. Okay, the enhancer can be, as you know, can be quite far away from the actual promoter or the transcription start site, and but they are brought together. The enhancer bound activator and the promoter bound RNA-pol2, they are brought together by the mediator complex. To put it in perspective, and there you see that this SRB 8 to 11 are excluded, and then you can have another non-productive mediator complex where SRB 8 to 11 are involved, and it is recruited by a repressor. It is no longer an activator, it is now a repressor that recruits it, and then that excludes any interaction with the RNA-pol2 and the basic transcription factors. Okay, like the GTFs, the general transcription factors. Now, so for all this work, Roger Kornberg got his Nobel Prize in 2006. Father got it earlier for DNA polymerization. Son got it for RNA polymerization. Right? So just to get back to my original concept, original idea, that the promoter is not what is causing the variation, it is the enhancer. So what is it doing? What it is doing is that because of this, so whether transcription will happen or not, whether it will happen a lot or at a basal level or some intermediate level, is all determined based on the rate of recruitment of RNA-pol2 to the promoter. Now, RNA-pol2 by itself is recruited to the promoter, but the rate must be very low. Okay. Now how the rate is increased? So if you think about it, the rate will be increased if the concentration of RNA polymerase II near the promoter is increased. You get it? That a rate is definitely dependent on the frequency with which they will collide with each other. They will meet with each other, the promoter and the RNA polymerase II. So what this is doing is that the enhancer to the enhancer sequence, the activators are binding. And those bound activators will recruit the mediator. Yeah, so what I was saying is that the job of this enhancer and activators can be looked at. I'm not saying that that is the way, but the way I understand it, the way I find it more comfortable to logically look at it, is to simply interact with the mediator, the enhancer bound mediator, which in turn will interact with the RNA pol 2 and TF2D, the TAFs, to increase the local concentration of RNA polymerase in the vicinity of the promoter. And because of that, the rate of recruitment of RNA polymerase to the promoter will increase and the transcriptional activation will happen. Okay, because once it is recruited to the promoter, then the transcription happens by default. Okay, so that's how I look at it. Okay. So now, like you have been taught of all sorts of techniques for detecting the RNA. So if you want to know whether a particular RNA is present in the cell, you can do northern, you can do RT-PCR, etc. Whether you want to know whether, if you want to do it for a very large number of RNAs, you can do, colony hybridization or those kind of things. So these I'm not going to talk about again. This we have already. So now let us see where we are. So where we are now is that we can quite accurately determine abundance of an RNA. Okay, there is northern, there is RT-PCR, like quantitative RT-PCR, there is microarray, there is RNA-seq. There are many ways by which we can accurately determine the abundance of RNA in a cell. But what is still not achieved is the understanding that why a particular RNA is abundant in a cell type versus not in another. That is the regulation part. That what is not known is that is the relative abundance purely a reflection of the state that we want to measure. Like, are we seeing that this RNA is particularly more because it is a bone cell and it is not there because it is a liver cell? If so, then what are the reasons behind it? Though that is not clearly understood. And if we get the RNA abundance data, can we then use that information to understand the physiology of the organism or the life cycle of the organism, etc., etc. Those are the more complex things that we have not been able to so far completely understand. So, really the big ticket question that is out there is that how the cellular transcription machinery using a repertoire of RNA pol, the general transcription factors, the activated transcription factors and enhancers, how is it defining distinct subsets for distinct types of cells and doing it repeatedly without any mistake? Like every single human being will have the same kind of, more or less same kind of RNA abundance for a particular cell type. So that is the question that we will discuss in the next lecture. Okay, so far this one for this lecture. I'm sorry, there was a major emergency in someone I know, so I had to interrupt this lecture a couple of times, but I hope you will get the message. Thank you.